Welcome back, students, to the botany class again. Okay, so in continuation with your cell biology, today I would like to discuss with you a new cell organelle that is the nucleus. Okay, probably you all have heard about the term nucleus and we all know about it. Okay, let's see in detail the structure or the ultrastructure of the nucleus. Okay, this nucleus, it was first discovered by Robert Brown in 1831. It was first discovered by Robert Brown in 1831, and a true nucleus or the membrane-bound nucleus is present only in eukaryotic cells, while in case of prokaryotic cells, one of the defining characters is the absence of nuclear membrane. So the genetic material in case of prokaryotes, example of which we have studied the bacteria, the genetic material or the chromosome, which consists of a single secular DNA, it remains in the cytoplasm in a region known as the nucleus. So in prokaryotes, the chromosome is naked or it's without a nuclear membrane, while in case of eukaryotes, a nuclear membrane is present. As such, there is a true nucleus. So one defining character between prokaryotes and eukaryotes, which we already know. Okay, so the nucleus, it was first discovered by Robert Brown in 1831, and typical structure of a nucleus consists of nuclear envelope, the nucleolus, chromatic network, and nucleoplasm. And in the last class, we have seen that this rough endoplasmic reticulum, the endoplasmic reticulum which are associated with the ribosomes, they are found attached to the nuclear envelope or the outer membrane of the nucleus. Okay, the nuclear envelope, this envelope is similar to the plasma membrane again. So it's a double membrane structure. It consists of an outer membrane and inner membrane, and the space in between the outer membrane and inner membrane, or outer envelope and inner envelope, is known as the perinuclear space. And this nuclear envelope is not continuous. No doubt it's a double membrane structure or it's a double membrane envelope, but the two layers or membranes are not continuous, and instead they are perforated by pores at regular intervals. So these pores which are present at regular interval on the nuclear membrane is known as nuclear pore. And at these nuclear pores, the two nuclear membranes, the outer and the inner, they come in contact. They are in contact with one another. And these nuclear pores are the pores which occurs on the nuclear envelope, which helps in the exchange of materials between the nucleoplasm and cytoplasm. Okay, so nuclear envelope, double membrane, outer and inner, the space in between the two is known as perinuclear space, and they are interfered or perforated by nuclear pores at regular interval. Okay, so this nuclear pore is a complex, it's not a simple structure, but in fact it is a complex structure, and it is guarded by nuclear plug. So these are this nuclear pore, they are guarded by nuclear plugs, and together with nuclear pore, the nuclear pore and the nuclear plug, they constitute the pore complex. So it's not a simple structure, but it is a complex structure, and it is through these structures, or it is through these pores, that exchange of materials takes place between the nucleoplasm and cytoplasm. For example, as you go up higher, you will learn that this synthesis of mRNA, or during the process of protein synthesis, the mRNA is synthesized from the nucleus, and then it's transferred into the transferred to the cytoplasm, where it's converted into the message is converted into proteins. So this, the way that it moves out of the nucleus is through this nuclear pore. Okay, so one function is that exchange of materials. Then the other part of the other structure of or component of nucleus is the nucleolus. So this nucleolus was first discovered or was first observed by Fontana in 1874, and they are observed only in the interphase stage. You know that a cell cycle consists of interphase and a division phase. So during the division phase, this nucleolus disappears. disappears, and they are observed only in the interphase stage of the cell cycle. So during the division phase, this nucleolus, the contents of it go and becomes associated with the chromosome. The contents of the nucleolus also contains genetic information, which of course that genetic information is for the synthesis of RNA. So the contents of it go and becomes associated with the chromosome in the secondary constriction region, and it forms the satellite. So it forms the nuclear organizer, and so this they are observed only in the interphase stage, and this nuclear organizer is observed in 5 pairs of chromosomes in human cell. You know that a human being has got 23 pairs or 46 chromosomes. So this nuclear organizer, where the nucleolus becomes associated with the chromosome forming the Sept chromosome, satellite chromosome, they are observed in 5 pairs of chromosomes, and these are present in chromosome 13, 14, 15, 21, and 22. So Sept chromosomes or satellite chromosomes, they are observed in these 5 pairs of chromosomes. And this nucleolus is chiefly associated with the synthesis of ribosomal RNA. There are 3 types of RNA: messenger RNA, which is synthesized from the DNA, the genetic material of the cell; then ribosomal RNA, which is synthesized from the nucleolus; and transfer RNA, which is tRNA, which is formed as a result of modification of the polypeptide, which is synthesized from the nucleus again. Okay, so it's chiefly concerned with the synthesis of ribosomal RNA, and it's observed only in the interphase stage of the cell cycle. So nucleolus, and the other component is the nucleoplasm. Nucleoplasm, it's also known as the nuclear sap. It contains ions. The nuclear sap contains ions. It contains enzymes. It contains nucleotides and proteins. So the nuclear sap or the nucleoplasm, it's transparent, it's semi-solid, and it has low viscosity. So this is the material which is present, which is enclosed by the nuclear membrane. Okay, so the nuclear sap contains ions, enzymes, and nucleotides. Within the nucleoplasm, chromatin network and nucleoli are suspended. Okay, so nucleoplasm, it's transparent, semi-solid, low viscosity medium, and it contains ions, enzymes, and nucleotides and proteins as well. And within the nucleoplasm, the chromatin network and nucleolus or nucleoli plural, they are suspended in it. Okay, the next structure is the chromatin network. The chromatin network, they are uncoiled, distinct thread-like structures, and this chromatin network consists of DNA and basic proteins. They are called also as histones. And these histones, which are associated with DNA, they are basic, and they are particularly arginine and lysine. And how is these basic proteins? They are mostly positively charged. They are associated with this DNA, or these basic proteins are associated with acidic DNA. This DNA, they are negatively charged because of the phosphate group. So these positively charged basic proteins, or these basic proteins, they remain associated with this negatively charged. And these are known as histones. They are also known as histones, and particularly of the amino acid arginine and lysine. So this chromatin network, they are uncoiled, they are distinct and thread-like structures which are visible during the interphase stage of the cell cycle. However, whenever the cell is about to enter into the division phase, the cell will not remain in the interphase stage forever. It enters into the division phase where it undergoes mitotic or meiotic division. So when it enters into the division phase, this chromatin network becomes highly condensed. They become shortened, they condense themselves, and they become thick thread-like structures. They become thickened as a result of condensation and thickening. So when the chromatin networks condense and they become thickened, it is visible as a chromosome. Those structures, they are known as chromosomes. So chromosomes are highly condensed form of chromatin. So chromosomes are highly condensed form of chromatin, and the best to observe these chromosomes is during the metaphase stage of the cell cycle. And they were first observed by Hofmeister in 1885. So this chromatid network, they are in the interphase stage. These chromatid are uncoiled, distinct, and thread-like structures, while during the division phase, they become shortened, they become condensed, and they are called as chromosomes from there on. Okay. And so these chromosomes, they consist of DNA and histone proteins, and DNA being negatively charged because of the presence of phosphate group, they are associated with basic proteins, particularly adenine and lysine. So these proteins, they are polypeptides. We know that proteins are polypeptides, and so these basic proteins which are associated with DNA, they form a histone octamer. The 8 molecules of these histones, they form the core, and around which the DNA is rounded. This forms histone octamer, and the DNA is wound around that the 8 molecules of proteins, and so that structure is known as nucleosome. So nucleosome resembles like beads on a string. So it's like the necklace or beads on a string. So there are numerous nucleosomes arranged along the strand of the DNA. Okay, so let's see the structure of chromosome. So now this structure of chromosome, which is best studied during the metaphase stage of the cell cycle, has the following structures. It consists of centromer, it consists of chromatids, it consists of chromonema and chromomers. So what is centromer? Centromer is also called as primary constriction, and during cell division, the spindle fibers are attached to the centromer. When a cell starts entering into the division phase, the centromers, the spindle fibers, they are attached to the cell at the centromer and helps in the movement of the chromosomes. And each chromosome, before entering the division phase, the DNA replicates. The DNA replicates, and then from one it replicates double the amount of DNA. So, and as such, this chromosome consists of 2 arms. Okay, 2 arms, and these 2 arms, they are known as chromatids. These 2 arms, they are known as chromatids, and the 2 arms of a chromosome, they are known as sister chromatid, each of which has a DNA which has been doubled or replicated before entering into the division phase. Okay, so these 2 arms are known as the sister chromatids, and the centromer, where the spindle fibers are attached, and so these spindle fibers helps in the movement of the chromatids from one another. Okay, and the position of the centromer in a chromosome varies. They are not constant in all the chromosomes, but the position of the centromer varies in different types of chromosome, and accordingly, there are 4 types of chromosomes depending upon the position of the centromer. These are metacentric chromosome, then submetacentric, acrocentric, and telocentric chromosome. So this metacentric chromosome are those chromosomes where the centromer is present in the middle of the chromosome, which occupies the middle region or middle and the center position, where it gives rise to equal arms on both sides. The arms are equal. Okay. And submetacentric is that the centromer is lies a little away from the center, so that one side of the arm, side of the chromatids are slightly longer, and while the other is slightly shorter. And acrocentric is more towards one end of the chromosome, where the length of the arms become more significant, that one is shorter and one is longer. And telocentric, where the chromosome, the centromer occupies, it's at the one end, so that there is so that one side of the arms, they are considerably longer, while the other arms, they are shorter. So these are the 4 types of chromosome depending upon the position of the centromer. Another is that structure is this chromonema. So internally, each chromosome consists of single long piece thread of DNA and called as chromonema. So in each of these chromatids, there is a single long piece of thread, and this long piece of thread is known as chromonema, or long piece of DNA is known as chromonema. Other structure is the chromomers. These are highly condensed proteins of chromonema. The chromomers are the highly condensed portions, not proteins. The chromonema are the highly condensed portions of chromonema. And then secondary constriction and nuclear organizer. So this is the region where the nucleolus becomes associated with the chromosome forming satellite chromosome. The contents of the nucleus becomes associated with the chromosome at the secondary constriction region, giving rise to satellite chromosome. And this type of chromosome is, as we have said, discussed, it's seen in 5 pairs of chromosomes, 13, 14, 15, 21, and 22 of the human chromosomes. Okay, so what is the function of the nucleus? You know that the nucleus contains the DNA, and so it is the storehouse of genetic material. It contains all the information about the phenotypic characters, or the type of cells to be formed, or the type of the phenotypic characters to be formed. All this genetic information are present in this DNA. And internally, if you go at the molecular level, you find that this DNA, they are long polynucleotide chains, and they contain bases, and the genetic information on the DNA, they are translated into proteins. They are translated into proteins, where these proteins will determine the phenotypic character of an individual. The genetic information coded in the DNA, they will be translated into proteins, or into the language of amino acids particularly, and then these amino acids, which are the monomeric units of polypeptide chain, they determine the phenotypic character of an individual. So genetic information from the parent to the offspring takes place by the replication of this DNA, which takes place before the division, during the interphase. Before the cell enters the division phase, the DNA replicates, doubles the amount, so that half of it goes into one cell and the other half goes to the other cell again. So this is how transfer of genetic information takes place. So DNA, main function, storehouse of genetic material, and it's also the site where this mRNA is synthesized, and this mRNA carries the information where they are taken to the ribosomes, where genetic information is decoded into proteins, which ultimately determines the phenotypic character of an individual. And they are associated with ribosomes. They are associated with endoplasmic reticulum, forming rough endoplasmic reticulum, and these are also associated with the synthesis of proteins. So mRNA synthesized from the nucleus, they come out of the nucleoplasm through the nucleopore into the ribosomes, where the message is translated into proteins. So these proteins synthesized, they undergo modification in the Golgi complex, where they undergo glycosidation, or formation of glycoproteins and glycolipids, and then where they are transferred to the other parts of the cell. So all the cell organelles, they work in coordination. They do not work in isolation, but they work in a coordinated manner, where the function of one is related to the other. As such, the cell is able to maintain or sustain itself, or is able to maintain the individuality of the organism. So besides this, there are some other smaller structures like peroxisomes, glyosomes. So please go through all those in detail, but nevertheless, these are some of the most important cell organelles which we have discussed. So with this, I went up the cell biology, the unit cell biology, and in the next class, we can start with a new unit, the last unit, that is with physiology. So please come back as we start one of the longest and the most important unit of your syllabus. Thank you.